Hi, welcome to another exciting day of chemistry. Today what we're going to discuss is the reaction mechanism that occurs during glycolysis. So I have outlined a couple objectives that I have for you and you should be able to do by the end of this video. The first of these objectives is you should be able to define what glycolysis is. You should also be able to describe and identify the 10 reactions or the 10 steps that occur during glycolysis. And the final thing that you should be able to do is you should be able to identify the products that are produced at the end of glycolysis. So let's go ahead and get started. So we start out here with the basic definition of what glycolysis is. And glycolysis is simply a biochemical reaction in which a 6-carbon molecule is broken down into 2 molecules of 3-carbon pyruvate molecules. Pretty simple definition. We're going to get into the details of how that exactly happens. Some things to keep in mind during this whole process is that because glycolysis is a biochemical reaction, this reaction is specifically occurring in the cytosol of the cell. And we should also keep in mind that there's 2 distinct phases of glycolysis. The first phase is the energy investing phase, and those are reactions or steps number 1 through 5. The second phase is the energy generating phase, and those are reactions or steps number 6 through 10. So let's start out here with our first reaction mechanism or our first step of glycolysis. So a couple of things I want to point out before we get into the details of this step. Number 1, we start out glycolysis with glucose. That is this molecule up top here. And then out to the side, you will see these enzymes, and all these enzymes do is help move along the process of glycolysis. And specifically for this first step, the enzyme that's used is hexokinase. So we start out here with our glucose molecule. And the first step that occurs during glycolysis is phosphorylation. And for phosphorylation, what happens is that ATP is used to add a phosphate to our glucose. So all we're doing is we are adding a phosphate. And so when we add a phosphate, we end up producing glucose 6-phosphate. What I also want to talk about before we leave this slide is that this step number 1, acts as a control point. So the cell doesn't want to waste ATP, it doesn't want to waste energy. So what happens here is this enzyme here, hexokinase, is inhibited by glucose 6-phosphate. So what that means is when glucose 6-phosphate is produced, the hexokinase does not allow ATP to dump off an extra phosphate. Basically, it saves energy for the cell. So let's go ahead, let's move on to the second step of glycolysis. So for the second step of glycolysis, what happens here is we start out with this glucose 6-phosphate. And we don't add anything to the reaction, we just undergo an isomerization process. So during step 2, we undergo an isomerization process. And all this means is that the atoms involved in our molecule are rearranged. And so what happens is a difference in between these carbons. So what happens is this carbon breaks off of that ring structure there of glucose 6-phosphate, and what we end up producing is fructose 6-phosphate. Now we only have a 5-carbon ring there. And so we move on to step number 3. So for step number 3, again, we start out up top here with fructose 6-phosphate. And once again, we undergo our phosphorylation. Or we use ATP to add another phosphate to our fructose 6-phosphate. And again, all phosphorylation is we are adding ATP. So when we add ATP, or excuse me, I should correct myself here, we are adding a phosphate, not ATP. We are adding a phosphate to our fructose 6-phosphate. And so when we do this, what we end up with is fructose 1,6-bisphosphate. And so now we can go into steps 4 and 5 of our glycolysis reaction. So steps 4 and 5. We begin here with step 4. For step 4, what happens here is that we cleave or we break apart our fructose 1,6-bisphosphate. So step number 4 is what we call cleavage. Or essentially what we are doing is we are breaking apart our molecule that we start up here. And so specifically, we are breaking apart our fructose 1,6-bisphosphate right here where I drew this red line. And so what happens is that we end up with 2 different products. Now, the product that we have here on the left is dihydroxyacetone phosphate, and the product that we have here on the right is glyceraldehyde 3-phosphate. Now, if you notice here, this molecule contains a ketone. And because it contains a ketone, it's very hard to go through a reaction. So what happens here during this process is the 5th and final step of that energy investing phase, and that is step number 5, where again, we get that isomerization process to occur again. So what happens? Because again, this ketone doesn't want to react, we go through an isomerization process and simply the atoms rearrange where they're connected to or in the molecule, and we end up with 2 of these glyceraldehyde 3-phosphates. So right here at step 5, we have ended our energy investing phases. Remember, we've already invested 2 ATP into this molecule, or we've already utilized 2 ATP. So for steps 6 through 10, we're going to get some energy in return. Something to keep in mind is that we start out steps number 6 through 10 with 2 of these glyceraldehyde 3-phosphate molecules. Let's go ahead and get started with that energy producing phase of glycolysis. So, again, we've gone through that isomerization process, and what we have up top here is that glyceraldehyde 3-phosphate. Now, something to keep in mind throughout the next 5 steps is that we have 2 of everything that is produced. And the reason that we have 2 of these is we went through that isomerization process and we end up with 2 glycerolaldehyde 3-phosphates. So everything you see produced, we actually have 2 of because we started out with 2 of these molecules. So what we do here in step number 6 is we go through an oxidation process, or we oxidize our molecule, and we add phosphate, or we go through that phosphorylation. So step 6. We oxidize our molecule and we undergo that phosphorylation. And for our phosphorylation, we add that additional phosphate through this item or this molecule known as P sub I. That's just adding a phosphate. That's what that's telling us. What we also do to help oxidize our molecule is we use NAD+, and that gets reduced to NADH and an H+ molecule. So at the end of step number 6 here, what we have produced is 1,3-bis-phosphorylase glycerate. So we're ready for step number 7. So for step 7, we start out here with our molecule that was produced in step 6. And again, we still have 2 of these. So with this, we are ready to get that payoff. We're ready to get that energy back that we invested. So for step number 7, what we are going to do is we are going to undergo, or we are going to have a phosphate transfer. And what that means for us is that we are going to make ATP. And so what happens here is that our molecule has ADP come by and pick up a phosphate group, and this produces ATP. Now again, remember we have 2 of these. So actually what's happening is we get 2 ATP out of this process. Now what we're left here when we take off this phosphate group is 3-phosphoglycerate. And that's where we start here with step number 8. So we start with our molecule that we ended in step number 7. And so for step number 8, what happens here is we go through yet another isomerization process. So we undergo another isomerization process. Again, the atoms just switch, or they rearrange the places in which they exist here in the molecule. And so we end up at the end of step number 8 with this molecule here known as 2-phosphoglycerate. So we start out step number 9 with our 2-phosphoglycerate. So for step number 9, what we do is we undergo a dehydration reaction. So what happens here is this alcohol, this OH group here, what happens is it undergoes dehydration through the help of this enzyme. And when this happens and we get that dehydration or that water pulled off this molecule, what happens here is we end up with our phosphoenol pyruvate. We're getting pretty close to producing those 2 pyruvate molecules at the end of this process. So we go through step number 10. So for step number 10, what happens is we go through yet another phosphate transfer. Essentially what we're doing is we're making ATP again in this process. So for step number 2, we go through a phosphate transfer. And essentially what that's saying is that we are producing or making some ATP. And now remember, again, we have 2 of everything in this energy producing stage because of that starting material we had 2 of in the energy producing phase. So we've actually made 2 ATP just in this step alone. At the end of this, once we take this phosphate and we make that ATP, we get to our final product here. And this final product is pyruvate. And so we have reached the final step of our glycolysis reaction, our glycolysis mechanism. And so we can talk about the products of glycolysis. So remember, because we started out with 2 of the glyceraldehyde 3-phosphates, we actually end up producing 2 pyruvate molecules. And through this process, we have produced 4 ATP. Remember, we had 2 steps that produced ATP, but we have to double the amount because we started out with double the amount on this energy producing state. And then in addition, what we've also produced is 2 NADH and 2 H+ molecules. And that was from that step number 6 where that was produced. So, as we get to the end here, I want to ask a couple pause and predict questions. And so, these pause and predict questions involve both the energy investing phase and the energy producing phase. So the first 2 questions here are going to emphasize, or going to ask about that energy investing phase. So I want to know, what is the name of that enzyme used in the very first reaction of glycolysis? And the second thing that I want to know is, I want to know the 2 products that are produced after the 4th reaction of glycolysis. And to help you out a little bit here for these pause and predict questions, I have all of the energy investing phases of glycolysis. So that first question, I want to know what enzyme was used in this very first reaction. And this second question, I want to know the name of both of these products that are produced at the end of step number 4. So take a moment here and pause the video and answer these questions, please. The second set of questions that I want you to have for our pause and predict questions involves the energy producing phase of glycolysis. So I want to know what coenzyme is used in the 6th reaction of glycolysis. And question number 4 here, I want to know what is this coenzyme reduced to in that 6th reaction of glycolysis. So to help you out here, again, I outlined all of the steps of glycolysis here. And what I specifically want you to identify is what is the coenzyme that should be placed right here, and then what is that coenzyme reduced to. So go ahead, pause the video again and answer questions 3 and 4. All right, hope you've enjoyed this video and hope there's more to come.